So what is PDV vector? PDV vector is program data vector. So how is the data SAS data step processed? It is processed in two phases. One is called the compilation phase and the other is execution phase. So we'll be learning what will be running in the background for the compilation phase and what is happening in the execution case. So what will be happening in the compilation phase is first the dataset is read in the background, and in the execution phase the observation is outputted into a dataset. So in the first step the SAS dataset reads each observation, and in the second step it executes that observation into a dataset. These are the two steps that has been followed to create a SAS dataset. So we'll be learning, and for each dataset, uh, the step is in the background. Two automatic variables are created. One is underscore and underscore. The other is underscore error underscore. Let's take an example and see how it works. I'm creating these two variables called underscore and underscore error underscore are defaultly created in the background. So if you want to see what are those variables and what will be the values for these default automatic variables, so I'm creating new variable called num, and I'm printing whatever is running in the underscore and underscore, and I'm creating other variable called errors under value, and I'm trying to see what is printed in the automatic variable underscore error underscore. So if I run this, so what does underscore and underscore do? It counts the each observation. See what is printed here, it counts each observation. So the first observation is compiled and then it is executed. Once the first observation is compiled and executed, it goes into second observation. Once second observation is compiled and executed, it goes into third observation. So on it goes into 19. It also starts compiling phase for 20. So let's say we have 5 observations. Yesterday we discussed when we are creating do loops, right? When we are creating do loops, if we have 3 variables, why it is reading till 4? Because it tries to read for one extra variable. So compilation phase, if we have only 3 observation, compilation phase will run till 4, but fourth variable or observation, whatever it might be, let's say we have only 3 variables and 3 observations, compilation phase is running for the next fourth observation also, but it is not executed because we don't have results. Similarly here, how many observations do we have? 19. In the background, compilation phase has been run for 20th observation also, but it is not executed because we have only 19 records here. So this is how each record is counted in the background, and if we have any errors will be printed as 1. We don't have any errors, so all the compilation and execution phases are running good. This indicates all the compilation and execution phases are running good. So if we go into detail, I have used infile statement or cart statement to execute this dataset, but I had a missing value here. So how many observations will be created here? Any one of you? Totally we are expecting 3 observations. Okay, let's see. Test. The third observation is not executed because we have an error. So if I go to log and check it, so what is the third observation I am expecting? The first observation, Vikas, his age is 22, he is male, and his scores for math, science and social has been printed. 99, 98, 90, 99, 98, 90. So, and the total is calculated because we have used here total is equal to this one. Similarly, second observation is also created. But what is happening with the third observation? Okay, name is read as Vimala, age as 21, gender as female. We have maths and science scores, but when it comes to social, we have score missing. So we have num and error value missing because we got an error here. So SAS reads error as numeric 1. And for which observation has this error occurred? For the third observation. So that's how it is read in the background. So if you go more into more detail of this, let's take an example called start dataset, and we have here 4 observations and 2 variables. Now if I want to select last observation of variable z, what will we do? Monica, what will be we doing if I want to select this record and this record? Issue here. Monica, if I want to select these 2 records, what if I want to select these 2 records, what will be I doing for that? Vidisha, what will be I doing if I need to select the last records for Z? If I want to select last A and last BB. Do you guys have some answer for that? Vidisha?
Um,
Yeah, go ahead, Faisal.
can we try group by and use the last, like n operator n underscore n underscore a variable to get the last variables out?
I'm just asking, I want to select simply last record of each category of Z. Last record of each category of Z. It's simple, guys.
You have to sort by descending order and then.
And just sort by Z, and what's next step?
And then if you select one, you need
Last
to
two.
change the first one.
Don't compare it with PDV vector. This different example. I don't want this to compare with my PDV vector example. You are trying to compare this one and trying to implement that to this dataset. I'm not expecting that. I'm expecting to select last record. All we need to do is we need to sort it and select last dot, right? Last dot of Z selects this record and this record, right? See, sorted by Z. It's anyways already sorted. No need to use proc sort again. So if I use last dot,
G. Good.
so we got A is equal to 8 and B is equal to 4. That's what I'm expecting, right? Where is the other one? So A is equal to 8 and B is equal to 4. I'm only expecting those 2 records. Okay, now I'm multiplying it by 2 and I'm selecting last dot. So my results are 16 and 18. So how is this running in the background? If I want to check what is happening after each and every observation, what is happening after the first observation, how is the compilation phase and execution phase done, and what is done after second observation, and what is done after third and fourth observation. If you want to go in detail with that, I can use put to print each of my statements. I'm printing into 4 different statements. I'm printing my first, second, and third, fourth. Oh, I'm printing each compilation phase. Let's go to log. This is how it is run in the background. Let's copy this to our window. So for the first observation, always SAS considers first dot and last dot as 1. So here we are sorting it by Z, right? So my first dot and last dot will be defaultly 1. Let us concentrate only on the first observation for now. So the compilation phase has been started. It considers first dot and last dot as 1. When it comes to error 0, n is equal to 0. And there is no result printed yet because still it is in the compilation phase. And when it comes to second round, again, it in the first observation Z is also not read as AA. This is a simple first line that is run without reading any observations. It only takes first dot and last dot and it looks for any errors. Okay, then it goes into second compilation phase where it reads the value called AA, and my Q value is 5, right? The value for first observation is AA, and when it comes to variable Q, my value is 5. And here my first dot will be 1, last dot is not 0 because I still have one more observation. So this is not ending here, so my last dot will be 0. Still D is missing. It is not printing the result yet because it is only reading till here. When it comes to third step, it is trying to read the result here. In the first step, when this is happening, data result, this line is read. When it comes to such statement, it is taking the first observation in the first compilation phase. When we are going here, that means when we are going to by statement, and when we are going to here, it is printing D is equal to 10. 2 times of 5 is 10, and still this is not my last dot, right? So it goes into one more compilation phase. So here my first one is ending. Here my first observation is ending, and this step is the execution phase for the first one and compilation phase for the second observation. Here two things are going on. So why, even though you see n as 2 underscore n underscore as 2, that means it is reading the second observation, but still it executing the first observation. So still we have values a and a. Q is equal to 5. This is my first dot is equal to 1, last dot is equal to 0, and result is not printed because the result is already taken here and it is just executing here. My first observation is executed and second observation's compilation phase has been started. Now again it starts with the second dot. This is my last dot for AA, right? So this is my first dot for AA and this is my last dot for AA. So what's happening here? So last dot is 1 here from here. For all the second observation, my last dot is 1. For all the second observation records, my last dot is 1. Here it is not 1 because still it is executing the first observation. So my last dot is not 1. It is just compiling the first observation. It is compiling the second observation and executing the first observation. So on it continues to observation 3. and observation 4. And why do I have observation 5? I have only totally 4 observations. Why it is going for fifth record? Because it is trying to go for fifth observation. It is executing the fifth observation, and no, sorry, it's compiling the fifth observation and executing the fourth observation in this line. What is happening is it is compilation phase is going on for fifth observation and it is executing the fourth. Fifth observation is not executed because we have only 4 records totally we have 4 records. That's how it is run in the background. If you want to see that, we are printing it stepwise. What does put do? Put prints into log. So if you are getting any errors, we should know where it is coming from. Is it coming in from the execution phase or in the compilation phase? So I'll send you this document. You can follow this. So what is happening? A data step is processed in 2 phases. First it is compiled and then executed. And this is the example I'm explaining you. And if you want more detail what is happening, you can go through this PDF. It explains line by line what is happening. So the data step is impiled in a loop when we are doing input, set, merge or any statements. That is first called compilation phase. Then SAS execute the statements in the order line by line. So each observation by observation or record by record for each observation in the SAS dataset being read. And then underscore and underscore counts iteration. So each observation is counted as, these are called my iterations. So these are my iterations for record 1, these are my iterations for record 2, and these are my for record 3 and for record 4. At the bottom of the data step, SAS by default writes an observation into a dataset. So that's how it is going. There are some more examples if you want to go in detail. All you need to know is how does the, if someone asks you how does the SAS data step runs or how are observations created, you need to know it is run by using PDV vector, and it is done in 2 phases. One is the compilation phase and the other is execution phase. And what are the 2 SAS automatic variables that are created? Underscore and underscore error underscore. That's all you need to concentrate more on. Okay. So that's all with the basis, and we'll be knowing what is clinical data and what is done actually in the clinical data. So is everyone from science background? Let me come one by one.
Nope.
Yeah. How about you, Anjali? Not Vidisha, okay. And Monica? No?
Pharmacy in chemistry.
Anjali, you're from chemistry?
Yeah, I
Uh,
did grants in chemistry.
okay. And how about you, Monica?
I'm from pharmacy, B pharmacy.
Nalini? Shravya?
Yeah, I'm from pharmacy.
Okay.
Yeah.
Can anyone explain what is the difference between phase 1, phase 2 and phase 3 trials? What will be going on in these trials? Whoever are from pharma background. Vidisha, are you from chemistry background or any science background? No, okay. Monica, can you explain everyone what is in phase 1, phase 2, phase 3 trial? Whatever you know about phase 1, phase 2, phase 3 trials, feel free and say it to everyone. Again, I will explain it to everyone.
No, Kumar.
No idea?
No idea. Like in phase 1, it researches the test and the new drug treatment in a small group. They researches the drug in a small group,
Mm-hmm.
like to evaluate its safety and also the side effects.
Okay.
And coming to phase 2, it
Mm-hmm.
is the treatment is given to a large group,
Mm-hmm.
and it is also again tested for the safety.
Okay. And how about phase 3? Yes, sure.
Phase 3, like it is given in large groups, same as phase 2, but here we will compare the effectiveness and will also monitor the side effects,
Yes. Yeah.
and
Uh,
will also collect the information.
you're right, but even in phase 2, there is a efficacy part. So in phase 1, there will be only safety part. So and these are done normally on group people ranging from this is the size of the group. And when it comes to phase 2, the size of the group varies from 100 to 300 patient, and the duration might vary from 1 to 3 years. Phase 1 can be sometimes few days or couple of months. And when it comes to phase 3, it is done on several thousands of patients. In phase 1, only safety part is done. In phase 2, both safety and efficacy. And when it comes to phase 3, only efficacy is done. And these are done to identify proper dosage ranges from investigation drug, toxicity, nutrient based and investigation tested for the first time in small numbers on healthy volunteers. Volunteers are administered to a drug. Sometimes they are asked in the clinic to see what is their safety. They're asked to stay overnight in the clinic, and patients are closely studied. And if patients pass phase 1 and if they think there are no side effects, then second step will be continued for phase 2. And in phase 2, apart from safety, efficacy is also tested in the phase 2. What are the side effects that are being caused, or is there any improvement in the patient after giving the drug? Is his is for whatever we are treating, is there any development in the patient? So those things will be checked in phase 2. And phase 3 goal is to we look what are the positive effects because of this investigational drug and negative effects, and we'll monitor this in a large group.
So in phase one,
Most Mhm.
I read somewhere that it's for healthy people only. They do just to check the side effects of the drug. So
Yeah,
it
they
basically.
will only take the safety part here. So they will check that is everything safe in using this drug, and they are not worried much about how is this drug acted, or what was the percent of curing for the injury or whatever. So those things will not be much concentrated, but is it safety, is it causing any side effects or not, those things will be tested.
So that what they need to do on healthy people so that
Yeah,
they'll understand better.
yeah. And when it comes to phase 3, when most of the drugs whatever reaches phase 3, those will get approvals most of the times, because they will do after phase 2 studies, they will do lot of analysis. They will, and in each phase, after completing each phase, they will be submitting to FDA for the approval. Once phase 1 is completed, they will submit all the data of the phase 1 patient's information, what are the side effects or anything, they will report to FDA. Once phase 1 is done, they will do the same thing for phase 2. but after phase 2 they will do lot of analysis, because in phase 3 big group of patients is involved and lot of money is involved. So they'll do risk analysis tests, all these things, to confirm that is this drug going to be successful or not, and then they'll go for phase 3. What is an informed consent? Let's say they want to start either phase 1 or phase 2 or phase 3 study on a group of patients. Let's say might be for phase 1 80 to 100, or for phase 2 100 to 300. Before starting these trials, they will decide to select how many number of patients, and they will explain the all the patients that this is the study and this is what we are doing for, and they will write an informed consent which is understandable by in a layman's language, so that patients who are participating in the study will be knowing on what they are getting drugs for. They will explain everything, and patients can anytime willingly they can drop from the study. They are not forced, like once you sign in the study you cannot drop or something like that. They can drop from the study anytime. So they will read all these things, like what are the procedures followed, what is the expected duration, a description of any reasonably foreseen risks or discomforts. All those things will be explained initially to the patients, and if they are interested, then they will be signing the informed consent. So before signing, the patient should understand why they are being participated for the study. So that's the informed consent. Once the patients are agreed that they're going to participate in the study, the informed consent is signed, and patients a date is given, like first dose date or screening visits date is given to the patients, and they will continue to participate in the study. That's what with informed consent. So in the clinical SaaS, we'll be working on concepts called CDISC concepts, CDISC, SDTM, and ADAM. What are the CDISC, SDTM, ADAM? Because when it comes to data, once the data is collected, let's say patients have signed the informed consent and they came to the clinic, and in the first visit is called like screening visit. The patient's blood tests, like lab tests, their ECGs, their vital signs, and are they using any drugs, or are they having any other concomitant medications that are using. All this data is collected. All the data is collected in a different data sets, like adverse events are collected in one data set, or lab tests are collected in the other data sets. And when it comes to lab, again there are chemistry tests, there are urinalysis tests, and there are some other microscopic tests. For the blood test, when it comes to again blood tests, there are hematology tests, RBC, WBC tests, like this. There are a lot of tests will be going on. When it comes to vital signs, they will be checking heart rate, systolic blood pressure, diastolic blood pressure. When it comes to ECG, they'll be collecting different ECG reports. So on, they'll be collecting all these results, right? And the results will be collected in different data sets. But when all this data is submitted to FDA, different companies collect the results in a different way, and they keep the variable names in a different way, and their control terminology is different way. What is control terminology? Let's say in some country some research is being done in US and also in UK. Let's say in US they call headache as headache, and let's say in some other country they call it as soothing pain. There is different terminology for all these adverse events or these, right? So FDA don't want to make things complicated, and they have started one rule book called CDISC rule book, and there they defined how to create each data set. They have mentioned how to follow rules here. Let me open CDISC. So in CDISC again we have SDTM and Adams. In the SDTM datasets they will be defining how to create each variable and how to keep the observations and their values. See, they have defined all the data sets into these groups like special purpose domains, interventions, events, findings, trial design domains, and relationship data sets. In these groups again, let's say adverse events will be going into this data set. All medical history, previous medical history, like does the patient have any surgeries or does he have any problems, all such kind of information will be going into medical history dataset. All lab test information will be going into lab dataset. ECG results will be going into EG dataset. Vital signs will be going into VS dataset. Let's open lab for example. So in the lab again, these are the variables that should be present in the dataset like study ID, domain, new subject ID, sequence, what are the lab tests, what is the category of the test, what are the results. These are the names of the variables in the dataset. So they have created standard rules how these variables should be, what are their type, like it should be a character variable or numeric variable, and what are the labels that need to be assigned, and what are the rules here. If you see cdisk notes, those are the rules that need to be followed in creating each of these variables. So we'll be going this into detail after we go with the CRF page. So okay, they are collecting lab results. Let's go to CRF page. What is CRF pages? CRF page is nothing but case report forms. So all the patient's information is collected on the CRF pages. So once the patient signed the informed consent and once he started coming to the clinic, and the study is divided into multiple visits, might be, let's say they have planned study for 30 days. They have planned study for 30 days and they have asked the patient to come at 5-day interval. So how many visits will be having? Totally one will be screening, and patients will, each patient will be having 6 visits, right? Let's say study is designed only for 30 days and patient was asked to come to the clinic at the interval of 5 days. Totally there will be 6 visits and one will be screening visit. In the screening visit, initially they will be taking all the results before giving our drug, because they want to know is there any side effects which are being caused, were there before start of the study, or those adverse events are causing after the study. So initially they will take reports without giving our drug. After giving our drug again, they will assign certain visits. They will assign certain visits and they will go step by step. And all these results or whatever data is collected from the patient will be entered in the case report forms. This is the case report form for one patient. So let's say our study has 24 patients. Here we have 90 pages, right? All these 90 pages are for only one patient. So similarly, we'll be having, if we have 25 patients, we'll be having 25 patients case report forms. So once the patient has signed the informed consent, on what date and time he has signed the informed consent, the date will be entered here. So site number, in which site the patient has signed the informed consent, and they will assign a particular subject ID for each patient. Subject ID is nothing but they will assign certain ID for each patient that will be entered here to identify who is the patient and whose information is entered in this case report form. So once this information of site number and subject ID is collected, once the patient signed the informed consent, the date and time will be entered here, and the format collected is date 9 dot format, right? This is the format that is collected in the CRF page. Once that is done, screening. In the screening, what is the color, what does they collect? Date of birth, what was the date of birth of the patient, and what is the gender. In this study, this is a male drug, so we are having only, in our CRF is designed in such a way that we are only having male. Sometimes they will be having both male and female. Whoever the gender is referring to, they will tick that column in the CRF page. Once gender is collected, their ethnicity and their race is collected. So, and the date and time on which the screening is done is collected. And what was his past medical history? Did subject report any past medical history? If we say yes, information should be filled here. Does he has any surgery, or is he going with any other treatments? Like, let's say patient has diabetes. Uh, what was the condition or illness, and is it ongoing, or it was an existed surgery? If you're talking about any surgery, is it still ongoing, and what was the condition or illness of the patient? If you say no, this page will be empty. If patient says I'm good, I'm completely good, I don't have any effects till now, so then if this page was ticked as no, all these columns will be missing. And when it comes to surgical history, if he has gone through any surgeries, that information will be here. Here we'll be having only any medical conditions. Here we have separate page for surgical history and on what date and time it was collected. Then physical examination. He's all they will ask each part by part. Do you have any effects in neck? Do you have any pain? Do you have any cardiovascular problems? Do you have any lung problems? Do you have any problem with your abdomen? If it is normal, this column will be missing. If abnormal, we need to specify it here. What was the abnormality? If he says I have some problem with respiratory, what was that? Do you have any eosinophilia or something? If you have anything like that, if abnormal, the reason should be mentioned here, and on what date and time this was collected. Similarly, vital signs of the patient was collected. This is all for screening. You can see the visit is screening. Screening means before giving our drug. Screening. All these pages are for screening. So in vital signs, what are collected? Weight, blood pressure, height, heart rate, temperature, and respiration rate. And vital signs are again collected in supine position. Here vital signs are collected in sitting position. Here supine, that means sleeping position. Vital signs are collected again. And again they are collected in standing position. And this is also for screening. For screening they are collecting three different vital signs: sitting, standing, and supine position. Then they are collecting ECGs, heart rate, QRS, time intervals, QT, and what was the date and time collected. If they are not done, this page will be missing. And heart rate is collected in beats per minute. These are collected in milli per second. If it is normal, ECG interpretation is normal, then this will be missing. If it is abnormal, then we'll be filling what was the abnormality. And then lab tests are done, like chemistry test, hematology, serology, cotinine, drug screen, and urinalysis. And in each category, these are different categories of lab tests. In hematology, again, we'll be doing WBC, RBC, platelets. When it comes to chemistry, we'll be doing phosphorus, calcium, sodium. So when it goes to calcium, we'll be doing like 20 tests, like potassium, calcium, sodium tests, all different things. When it comes to urinalysis also, we'll be doing multiple tests. So these are the different categories. And on what day was the blood sample collected for doing this test? On what date and time? Eligible criteria. So now screening is done till here. Now on the day one, on the first day of drug dose date, what are done? Similarly, lab tests are done, vital signs are done. Again, vital signs pre-dose on the day one, before giving dose, they are doing vital signs, and after giving dose, they are doing again vital signs. On day two, on day three, day four, so on. All CRF pages contains entire information what is happening with the patient on each visit, and the results are collected in this CRF page. And these results are inputted into Excel files, then converted into SAS datasets. Vital signs, day 13. See, and vital signs are again collected hourly on day 13. If you see 5 hour post dose, 5 hour post dose, and again sitting, standing. Why we are doing so many vital signs? Because the CRF pages, this study is recording. There is a drug called Udinafil, and the effect of Udinafil. If let's say what is an Udinafil is a male steroidal drug. So what is the effect of Udinafil on doxycycline? So let's say patient has some blood pressure problems. If that patient who is using doxycycline drug, and our study drug is Udinafil, if he is using Udinafil with doxycycline, does the patient has any effect? So we are testing it on a, we are testing some drug interactions. We are testing our drug called Udinafil on doxycycline. As doxycycline is an blood pressure drug, we want to see what are the changes happening with the vital signs. That's why we are collecting so many vital signs. If you are doing any research related to blood, we'll be doing lot of blood tests. So that's why we have so many CRF pages for vital signs. See, 8-hour post dose, 7-hour post dose, sitting, standing, supine, lot of vital signs are collected. We'll be going into detail what drug is given. I'll be explaining protocol and SAP also. Day 18. Again, after completing all the visits which we have defined, initially when screening is done, again final visit is done. On day 30, they won't give any drugs. They will ask the patient to come to clinic after 3 or 4 days. Then they will do final visit analysis again. So then they will compare all these visits. What was there at the screening, and what was the patient's condition in all different visits, and what was his condition at the final visit. So they will do analysis at the final visit, end of the study. They will also collect adverse events. So on day 1, what was given? Doxycycline. So here this study is designed for 18 days. On all 18 days, patient was given doxycycline because that patient has some blood pressure effects. So patient is given with, patient who is taking doxycycline want to use our study drug Udinafil. Udinafil is an male steroidal drug, and is that drug affecting doxycycline? So that was our study purpose. So, and when was our study drug given? On day 13 and 18. So patient will be taking regularly his drug, and on day 13 and day 18 will be giving Udinafil, and will be testing the effect of Udinafil on doxycycline. That was our study goal. We'll be also testing concomitant medication if patient is using any other medications. What medication is he using, what was the dose, what was the frequency, and when was this collected. Is it still ongoing, or did he use previously? So if patient need to participate in the study, again there is called inclusion and exclusion criteria. In inclusion criteria, we'll define some rules like the patient should meet all these criteria if he need to participate in the study, and if he is doing any of these, he is excluded from the study. So patient, some inclusion criteria will be like patient was not supposed to smoke, patient was not supposed to take alcohol during our study, patient was not supposed to participate in any other research when he is involved in our study, or he was not supposed to take any drugs without our notice, because those drugs might affect or cause those adverse events. So whatever, when he is in our study, he should intimate us if he is using any other drugs. And if there are females in our study, we should not supposed to take pregnancy patients until unless we are doing research on pregnancy or some drugs like that. So these are different inclusion criteria that need to be followed, and if patient is having any of these, he is excluded from the study. Those are called exclusion criteria. So this is about case report forms. So what are the different documents we need? Once the study is started, they will design a protocol. they will design a protocol and they will decide how many number of patients should be selected, what was the doses that should be given at each visit for the patient. Who will be designing this protocol? Medical writers, statisticians, and study clinical sponsors. Who can be a sponsor? Any of the pharmaceutical company or any CRO company or an individual person who can fund for the research will be the sponsor for the study. So the sponsor will also be participating in creating this protocol. He might be creating it, but he will be reviewing it. So he should know, right? Because as he is a sponsor, he should know what is the study planned for, how many number of days, or how many number of patients should be selected. So such information is given to the sponsor, but all this protocol is designed by medical writers and stats or any PhD biostatisticians. So what was the study recording? What was the protocol title? Interaction of Sildenafil and doxycycline in healthy male volunteers. Okay, this is our study number, and what was our drug? Sildenafil, and what were the different doses of the drug given? 1 milligram, 2 milligram, 4 and 8. So for the persons involved in creating this protocol, medical writers, clinical pharmacology persons, biostats. Introduction. So our study drug is Sildenafil. What is Sildenafil? Is a new investigational drug being developed in the United States for treatment of erectile dysfunction. So this drug is used for erectile dysfunction, and this drug is similar to Viagra, which is released by Pfizer. So they are trying to release Sildenafil in competition with Viagra, and what they want to test effect of this drug on doxycycline. Let's say a patient is using a doxycycline. What is doxycycline used for? It is an blood pressure lowering drug. Doxycycline is a blood pressure lowering drug. Doxycycline is non-specific alpha adrenergic antagonistic indicated for the treatment of benign prostate hypertrophy, for the treatment of hypertension. So this doxycycline is used for hypertension. So patients who are having hypertension will be taking this drug every day. And if this patient want to use our study drug called Sildenafil, so is our study drug interacting with this doxycycline and causing any side effects? So that's our goal. So how many? This is a simple phase 1 study, and this is a crossover study. What is the this study is a single center study. That means study is done at one site. Sometimes studies are done at 50 different states in US. Then we'll be having 50 sites. Sometimes it might be done on different races. Sometimes 50 patients might be selected from Asia, 50 patients might be selected from Europe, and 50 patients might be selected from US. So if they want to get approval in different countries, they'll be involving different region people or different race ethnicity people. And this study is as a simple phase 1 study. This study contains only 24 healthy male volunteers. And what is double blinded study? In a double blinded study, neither the patient nor the clinical sponsors will be knowing what was the drug given. So what is double blinded is neither the patient nor the sponsor will be knowing what was the drug given. If let's say they're giving doxycycline and sildenafil, and if patient is knowing which is which, he will try to say yes, I'm good, I'm good. Even they will try to manipulate the results, right? So they will give the drug and they will ask just the results. They won't say what drug is given. And okay, and in single blinded study, the clinical sponsor will be knowing what was the drug given. Patients will be not knowing what was the drug given. And placebo controlled. In a placebo control, what does it mean is Let's say they have selected 24 patients. Let's say they have selected 24 patients. Each patient will be given a placebo and a test drug. What is a placebo? Placebo is, it might be just sugar coated tablet without any active ingredient, and test drug will be sildenafil. So patient will be given both with placebo and sildenafil, but patient don't knows which is an placebo and which is an active drug. So he will not be knowing what is an, what was, some patients will, let's say they have divided into 30 visits, right? And for the first 15 days they will be giving placebo, and the next 15 days they might be given test drug. But in double blinded study, patient won't be knowing this information, when is active ingredient given or when is placebo given, or he won't be knowing. He will be knowing what is involved in the study for, but he won't be knowing what time what was given, because if they say this was the drug given today, he will be trying to manipulate the results or something like that. So that's the reason double blinded means neither of them will be knowing, and in the single blinded study patient will not be knowing that information. I'll explain later what is a two-way crossover study. All subjects will receive doxycycline for 18 days, but different doses. On day 1 he will be receiving 1 milligram, day 2 he will be receiving, on day 2 and 3 they will be receiving 2 milligrams, 4 milligrams for 4 days, 8 milligrams for 11 days. So totally 18 days doxycycline is given, and on day 13 and 18 our test or reference drug is given. What is our test drug? Test drug is sildenafil. What is our reference? Placebo. Placebo is nothing but sugar coated tablet. Any questions, guys? We'll be continuing tomorrow with this protocol. So what you should know, what are phase 1, phase 2, phase 3 trials, why are these trials done, and you should be knowing what is in case report form. Case report form is nothing but where the information of the patient is collected, and why patient has to sign informed consent. And you should also be knowing what are inclusion and exclusion criteria, and how is a protocol designed, and what are the different topics that are covered in the protocol. We'll be learning more about the protocol tomorrow, and I'll also be explaining what is an SAP. SAP is nothing but statistical analysis plan. So in SAP, what information is present, like uh, it contains all the information of the protocol, and it, apart from that, it also contains it. The word itself says statistical analysis plan. So here they will do all the statistics, and what tables need to be created, what are the different shells, or what are the different outputs that need to be created. All such information is present in the SAP. Apart from drugs information and patient information, see what reports need to be, what statistical analysis are required for our study. So they will give some sample shells, how to create our, or how to report our data. So this is how they wanted the data. They will use some sample shells, so we need to create the data accordingly in the SAP. So what does protocol contain? Protocol contain entire study drug information, patient's information, and what are the different doses given, and how many days the treatment is being assigned for the patient. All such information is present in the protocol. In the SAP, same information is present apart from this statistical analysis plan, like, so what are the results or reports that need to be generated for to submit to the FDA. Such all information is present in the statistical analysis plan. So we should know what is an CRF page, what is an protocol, what is an SAP, and then we will be going to CDISC SDTM. What are CDISC SDTM concepts? How to create this data when we are creating reports. We have to follow standard procedures in creating all the variable names, label names, lengths, and standard rules for creating variables. It should be uppercase or lowercase, and what are the control terminology, how the unit should be reported, all standard units should be reported. We'll be learning all those things in next Wednesday's class. So next Wednesday's class will be also more of introduction about protocol, SAP, and CDISC concepts. Then uh, we won't be having class on next Saturday and Sunday. And next week we'll be starting with working on CDISC datasets by following all the rules from CDISC implementation guide. So this is called CDISC implementation guide. Did I have it open now? So this is called STTM implementation guide. We'll follow these rules and we'll start working on datasets. That's all, guys. Anyone has any questions?